So, you know, horse genetics is really exciting right now. We have all the fancy tools that a lot of medical genetics, human medical genetics has. And we have lots of fascinating veterinary and biological phenomenon to study. So I've tried to select tonight a few topics that I think will be particularly useful, that are quite sort of interesting and entertaining, and in some places where we may have something that needs a little clearing up in the general audience. All right, so I'm going to start off with a quiz. We don't have a Zoom poll, but I want you all to kind of take a scrap of paper and write down your answer, and then maybe we'll just kind of see where it goes. How many genetic tests do you think we can use for the horse today? So this is specific to the horse. It's a poorly written question. You can tell. My students complain all the time. Way too vague. How many traits do you think we can test for today? Is it A, 5 to 10? B, 10 to 100? C, 100 to 1,000? Or D, more than 1,000? Has everybody got your answer? I can't necessarily see your papers to tell if you're cheating or not. So I will trust you on this. There we go. I see somebody's got their answer on their paper. Good job. You are indeed correct. It is letter C. And my students will tell you I overuse letter C on multiple choice. But it is around 100 to 1,000 that we can test for with varying degrees of certainty. As it is in science, you know, we still call gravity a theory. So some things are better supported than others. We have at least 60 Mendelian traits, and then we have a whole plethora of markers for other more complex traits. But this surprises a lot of audiences because while a lot of folks are very comfortable with coat color tests, they have not heard of the many other things that we can test for that are useful for our horse breeders. Okay. The other point I want to make is that genetic tests definitely get pigeonholed as something that you only do when you're trying to select a stallion or make a match between a mare and a stallion. But with an animal as long-lived and potentially economically stressful as the horse, I think it's key to keep in mind the other potential uses of genetic testing. In the clinic, clearly there's some utility in diagnostics, and even for some of our other traits, there's usefulness there in having more information. And on the flip side, that information informs management. So it may be that for some conditions, particularly a few that we'll learn about tonight, that genes are not the end-all be-all to the story. And once you have that information, you can use it to inform how you manage your horse and avoid some problems, or maybe try to improve on some strengths. Okay, so when we were discussing topics, one of the topics at the top of the list was a genetic condition called PSSM, or polysaccharide storage myopathy. And here we're going to start with the form of this condition called type 1. PSSM results in something that horsemen have called tying up. And indeed, this allele itself is quite old and probably explained a lot of the early cases of tying up that have been reported for hundreds of years, right? Classic tying up, it looks like you will see it as stiffness, an awkward gait. Horses are uncomfortable, so they're often sweating and stressed. Once they begin to get muscle damage from these episodes, you'll start to see brown urine. That is definitely a red alert emergency sign. And because of the pain associated, they will refuse to work. Now, genetically, we know that PSSM type 1 originates from a gain of function mutation in the GYS1 gene. And we talk more about modes of inheritance at the end of the talk, ironically enough, but I'll tell you right now in a little spoiler, PSSM is dominant, and it's what we call incompletely dominant. So that means that if you have at least one copy of the gene, you will see some effects of the allele. The incomplete part comes in with the number of alleles. So a horse with one copy will have some effects of the condition, and a horse with two copies will be more severely affected. It's we also sometimes use something called an additive model. This is a little bit more dominant than additive, but as you'll hear me at the end of the talk about, our adjectives are definitely shades of gray in genetics. Mother Nature has taught us not to make hard and fast rules. So this case is something that I've grabbed off the internet. This happens to not be a Hanoverian, as you might have guessed. But this horse was a young horse who had gone into training. He was very promising. You can see he is definitely of a sort of a continental warm blood type, we'll call him. And although his demeanor was very kind on the ground, when they started him under saddle, although he did really well at first, he later developed these behavioral problems. So I asked my students in class, if you saw a horse behaving like this, what would be your suggestions? Where would we go? And spoiler alert, I'm a geneticist, right? But my students often say, get his back checked, check him for back pain. They say, check his teeth, make sure he doesn't have tooth pain. Some of them say, get a better trainer or a better rider. You'll hear the whole gambit of situations. And they're not all bad suggestions by any means. But this particular horse highlights a lesser known consequence of the PSSM1 allele. In fact, that little guy, he is a heterozygote for PSSM, and a lot of our PSSM horses will, rather than expressing a tying up type episode, will have minor muscle pain that results in behavioral problems and issues during training, especially in horses who are crossed with draft lines. And this is why. So a study done just a few years ago sought to examine the allele frequency for PSSM. So newly discovered disease. They were excited to have found this genetic test. And then they started looking at the draft horses. And all of a sudden they realized that 2/3 of Percheron horses have at least a copy of this allele. And in the Belgians, it's not a whole lot better, almost 40%. And in our stock horses, 5 to 10%, give or take, in that range. But we don't often hear of draft horses having terrible tying up episodes. So how could the allele frequency for a terrible disease have gotten quite so high in our draft horses? I have to guess, but Valberg and our co-authors here suggest that there could be positive selection for this allele. It turns out that the GYS1 enzyme, when we have that gain of function mutation, creates a slightly different saccharide molecule that's harder to liberate out of the muscles to use as fuel. And the hypothesis here being that for draft horses and working horses who would go through that feast or famine cycle of working hard during, plowing and planting season, and then getting a little fat over the summer, and then maybe working hard during harvest, and then potentially having a hard time finding forage in the winter, but maybe for those animals, this could be an advantage because they're less likely to raid those saccharide, those sugar storages in their muscles, and more likely to save it for a rainy day. It's a hypothesis that's pretty tough to test, but it has some logic to it. And I think it certainly could explain why the frequency for this is so high. It is also a very, very broadly spread allele and is found among sport horses. So given that genetic testing is so inexpensive, I think that it's not a bad idea to keep this in mind when you have particularly if you have a cross-bred horse who has some heavy horse bloodlines, as most warm bloods do, that it could potentially be a source of some of those behavioral issues we see in the ring. Now, the other good news to this story is that once we understood which enzyme was impacted in the PSSM1 condition, that we could very specifically target nutritional strategies to help to alleviate the signs. So because these horses have a hard time liberating sugars, what we can do is shift them to utilizing fat more commonly as a source of energy. Fats do not give them the same type of trouble. They don't build up the sugars that result in the muscle pain. So in general, the recommendation from our nutritionist collaborators is that if you have a horse who has PSSM1, consider low carbohydrate diets. Traditional grain, generally not a good idea. Increase fat to restore those calories, and as always, keep them in moderate and continuous work and change their diet with caution. With these type of interventions, many PSSM horses can live very active, healthy lives. Now, recently, in our horsey news and on the web world, we have heard about PSSM type 2. This type of PSSM looks very similar to PSSM1 on histology. So the purple squares that you see on the right-hand side of the screen, those are muscles, little muscle biopsies. And on the right-hand side, you see an image of a PSSM type 2 affected animal, where instead of an evenly purple stain, you see these clumps. That's that oddly structured saccharide. They have many of the same type of clinical signs as our traditional PSSM1 horses, although it's not well documented, colloquially I hear it's more mild. I also hear some folks say that their horses don't respond to the traditional PSSM type 1 prescribed diet, though again, we have no consistent studies yet to document that. Many of our warm blood animals are particularly affected with type 2 PSSM. So if your horse is positive on a biopsy, it's often the case that the type 1 genetic test won't actually help you. And so I think additional research is needed in this area. The recent controversy around type 2 is that we do have some services offering a PSSM type 2 test. However, there is a very recent publication that examined this type 2 test as it's documented in the US patent and found zero association to PSSM type 2 diagnosis. It's hard to get more solid evidence than a paper refuting it pretty substantially there. So I cannot recommend some of the advertised PSSM type 2 tests at all at this point. Hopefully, we will get more research done here so we can get a verifiable type 2 test on the market. Okay, so talking about sport horses, I have to visit for a short while about fragile foal syndrome. It's actually one of my favorite topics right now, and not because it's controversial, but because I see some optimism there. Fragile foal syndrome impacts a gene called PLOD1, and PLOD1 is particularly important for connective tissue of many kinds, all the connective tissue. In this case, the lethal form of the disease is recessive, which we'll look at the end of our talk, and that means it takes 2 copies of the allele to see the lethal phenotype. The challenge here is that these foals do often survive to birth, and when they do, you have a very high economic and emotional cost. Sometimes, as we'll see, they're born with some fairly catastrophic clinical challenges. As you can see from these small skin sections, there's a very obvious problem with the thickness and strength of their skin. So the normal is on the bottom and the fragile foal affected animal on the top. Recent work has shown that not only do we see these foals that are born with some pretty catastrophic abnormalities, this one has some curvature of the spine. Some of them have large open defects in the abdomen or across their limbs. But they also found that we more frequently observe abortion and stillbirth in homozygous foals. So it may actually also be impacting our reproductive efficiency, not just creating these very scary, catastrophically deformed foals. And I think for that reason also warrants good surveillance on the part of the horse industry. Now, a group in Germany actually examined a fragile foal in Hanoverian bloodlines. And through some testing of modern horses and careful pedigree analysis, they tracked it back into a single individual and attributed the allele frequency in the Hanoverian to that FW line in that individual. Since then, though, some additional papers have shown that the allele itself is really far older than the FW line. So we really can't go on a witch hunt and go after any stallion in that group. What is likely has happened is that there have been some very popular and very talented sires from these lines, and they have probably drug along this fragile foal syndrome allele along with their popularity. And we'll talk about that more a little later on. This paper estimated the allele frequency at 14% in their sample. That's something. That's a challenge. It's not insignificant. The other thing that we've shown in recent research is that while it originally was coined warm blood fragile foal, it is in no way a warm blood disease. So in this paper here, it was observed in stock horses and in a Haflinger. These are very diverse horses. It's also been found occasionally in the thoroughbred, which is remarkable because the thoroughbred often touts itself as having relatively few genetic diseases. So they're not at all immune. But finding it in these diverse lines suggests that actually the allele's been around perhaps longer than the formation of many of these breeds. So what can we learn from other genetic diseases that were discovered far earlier than fragile foal? The fragile foal itself has been commercially available for more than a decade. It just only recently came to our collective consciousness after some high-profile announcements and media coverage. And if we're going to go back in time and look at other genetic conditions, I think one that is particularly important to study is the first one that we actually went after as geneticists. It's called HYPP and is found often in stock horses. HYPP itself results from a change in a sodium channel gene, so it's important for our electrolytes. It is also dominant, but we'll call it incompletely dominant because like PSSM1, 2 copies is more severe than 1 copy. What we see in a horse who is having an episode is muscle twitching or fasciculations, and those can progress to the point where they create problems with normal muscle function. And remember, your diaphragm is a very important muscle. So they can lose the ability to stand or collapse due to paralysis and develop respiratory failure in a severe episode. What also has become broad knowledge is that most of the stock horse carriers of the disease trace back to a stallion named Impressive. Now, you can almost sense I always get uncomfortable here because geneticists do not name names, and every study I ever do is fully confidential. But in this case, the AQHA and its membership has been very vocal about disseminating this particular identity. And it's true. Impressive was a very, very successful stallion. One estimate suggested that in 1992, when he was 24 years of age, that he potentially had 55,000 progeny. I don't know if I believe that number, but I'm not going to try to do the math myself. In any case, he was extremely prolific as a stallion, extremely prolific. So I like to point out that the disease status for Impressive really does not diminish his value as a stallion in any way. He wasn't defective. He actually was not the founder for the condition. It probably originated before his generation. But just like most humans, all horses carry their share of disease alleles. I've given this version of this slide for well more than a decade. And early studies that I quoted said that humans carry on average 3 disease alleles. I recently refreshed this, and data from the 1,000 Genomes Project in humans now suggests that humans carry on average 40 to 110 disease alleles. And there's no reason that we should think that horses are any different. So every Impressive stallion probably had 40 to 110 disease alleles in his genome. Most of those go unnoticed because few horses are as prolific or as successful or as commonly line bred to, right? So his popularity and then the practice of line breeding back to him created a situation that both increased the frequency of his alleles and then increased the occurrence of horses with 2 copies who are homozygous. So we can't blame impressive or popular stallions. It's going to happen every time we have an excellent stallion, because no breeding animal gets all the genes for excellence and doesn't get genes for problems. The other interesting thing is that in 1992, when that particular genetic allele was discovered, the authors did, of course, trace it back here in their own pedigrees, but they right away stated in the paper that development of the genetic test provided an opportunity for control or eradication of the condition, which I thought was very grandiose language. I'm not quite so brassy today in my publications, but they got this into a journal called Nature Genetics, which is pretty darn good in our industry. Okay, so another quiz. Get out your little piece of paper there. And some of you probably know the answer to this story, but for those of you who don't, I want you to sort of sit down and scratch your head. The geneticist just gave you a test and said you can now eradicate the condition. What do you think has happened in the last 20 odd years? Almost 30 years now. I'm going to have to update my slides. Has the allele for HYPP decreased, stabilized, or increased? Yeah, I see some thumbs up. You can kind of tell. I'm not really good at suspense, right? I'm no good at surprise endings. Yeah, and in fact, the allele frequency for HYPP has increased, and this can be quite surprising. You know, when the disease was first studied, about a third of homozygous horses died before they reached 5 or 6 years of age. So it is, you know, when they started making policies or considering policies, it was no small matter. So here are the numbers. This is a study from 2009. We don't have solid historical data, but in 1992, we estimated that about 22% of halter type quarter horses were actually affected. So it's not too far, not that bad. But by 2009, that number had risen to 56% of halter type quarter horses, which is, yeah, that's pretty effective on their part. They did a good job of getting that number up there. This is despite some late legislation coming from the HYPP membership, where they actually banned the registration of horses who were homozygous for the condition in 2007. By that point, the cows had left the barn. We were probably way well on to those higher numbers, but I think it does kind of point out that they did recognize at some point that they had a challenge. So when we talk about this in class, I always ask my students again, so why do you think this happened? Why do you think this happened? And they come up with a couple of suggestions. One is definitely market forces. So if in the late 90s you had a stallion, you know, most of our stallion owners are actually small holders. They have 1 or 2 stallions in the industry. Very few have large numbers. And that stallion is successful, and he is your bread and butter of your business. And you are faced with the choice of, you know, you discover his carrier status and you have a choice: geld him and protect the future gene pool, but destroy your livelihood, or continue to breed. You know, the horse industry is comprised largely of small businesses, and it's hard to make the long-term choice. And we need a solution for that for sure. The other thing is, just like PSSM, once we understood which sodium channel was involved, we have developed targeted management strategies, including a specialized diet and things like regular exercise and careful electrolyte balance to help to manage the condition. So these days, we don't have any good studies, but you almost never hear of a horse dying from HYPP. It does happen, but it's a rare occurrence these days. So I think we have alleviated some of the negative problems associated with HYPP just by getting better. That doesn't mean we've gotten rid of all of them, but we think we have some of them. The last possibility is this theory that there's, again, an advantage to the carrier genotype. So not the horse that has 2 copies and might have problems with disease, even if they're non-fatal, but the animal who has just 1 allele. We hear this a lot in the industry, that these animals simply are better. And when you think about the clinical signs, those muscle fasciculations, and what is considered ideal in a halter bred quarter horse, it maybe starts to make some sense. Some of you might remember the TV infomercial about the magic battery-powered belt that would give you a six-pack of abs while you sat in the lazy boy chair drinking your Coca-Cola. Well, this might work by a similar theory where those muscle fasciculations are sort of exercising individual muscle groups and improving the tone that this area of the industry finds very attractive. We have just one study that actually supports this hypothesis. In this very, very small study, they looked at about 30 horses, if I remember correctly, and they were all half siblings, and half of the family had the allele and the other half were clear of the allele. And when they compared the show records for this group of horses who had a fairly similar history of training and of opportunities to go to competitions, it's not a perfectly controlled experiment, but they did make some effort in looking for their case study group. When they looked at these offspring, those that had one HYPP allele consistently earned more points in the halter ring. So presumably, there is something that science has not detected in measuring the muscles or looking at them microscopically that judges of halter horses can detect and somehow find pleasing. Don't know what that is, but I think there's something there. We need to look at it anyways. So there's more to this story. All right. So what about fragile foal? We're going to circle back now. Let's go back to our earlier story. Well, we don't have 20 or 30 years of wisdom to draw on at this point, but we can look to human medicine. And in fact, there is a condition fairly similar to fragile foal in humans, and it might tell us a little bit more. This gentleman here in the photo, he's actually affected by this particular condition. It's called Ehlers-Danlos. It's a bit of a mouthful there. And there are many types of EDS-type syndromes. Gosh, I think there's 11. And one of those actually impacts the human version of the PLOD1 gene. Now, in humans who have two copies, they do generally live. They don't often die immediately after birth, but they do tend to have eye, spine, and quite notably for me, vascular disease. That vascular disease in these humans can manifest as aortic dissection, which is where your large blood vessel that runs from your heart will spontaneously rupture, and things like strokes. These humans also tend to be taller than average. They tend to have a slightly narrower chest or narrower build, and most interestingly to me, they have joint hypermobility. So the picture you see on the right, we can all you kind of see this and think about it. Take your own hand and take your thumb and try to touch it to your forearm the way that this image has. Now, most of you won't be able to do that. But if you're an individual with Ehlers-Danlos, you can do that without any difficulty at all. The human version is recessive, but it's also milder than the horse version. With two copies, they can lead a reasonably healthy life. But even in the human version, we can see that carriers, those who have only one copy, which would be similar to most of our living horse carriers, they have less than the normal amount of that PLOD1 enzyme, somewhere between 50 and 80%. And this is a very small sample size because it is a rare form of this disease in humans. So analogously, it seems likely that horse carriers of a PLOD1 change or carriers of fragile foal probably have less than the normal amount of enzyme. And I think it's worth study to see if that less than the normal amount impacts them physiologically. The other interesting thing about fragile foal was how it kind of hit the news and made a splash. You know, we had known about it for quite a while in the genetics world. I think that part of the reason this came to light was the introduction of broader testing panels. So certainly, I think that, you know, I've heard stories from some of our clinicians that they probably saw cases in stock horses, but never knew to consider testing for fragile foal because at the time it was called warm blood fragile foal. So that didn't help us out any. But I think once we stopped targeting testing only sick animals and started thinking about testing everything, then we got an unbiased picture of how many horses were potentially carriers. Also, having a few high-profile sick foals and people willing to share that broadly across the internet certainly captures, kind of pulls at our heartstrings when we hear about those terribly sick foals. Okay, So what have we kind of learned here from managing genetic disease? And then we might take a quick pause before I go on to complex traits, because I feel like we need to catch up a bit. So as we have learned from the AQHA, considering organization level policy some 15 to 19 years after discovery of an allele like this doesn't work very well if you want to control the allele. You know, so I think it's worthwhile to think about their situation and consider what can we do differently. I certainly think that it's the breeder's job to keep themselves in business, to be able to make ends meet and try to keep going, but it's the breed organization's duty to think about the long-term benefits. They have to keep the industry healthy now and in the future. That's a hard job. I see I've got good questions in the chat too, so we're going to come back to those. Now, while sires have an outsized impact on the gene pool, don't forget about your dams, and especially breeders who have just a few brood mares. I think that you in particular cannot afford the emotional and economic hit it takes if you have even one sick foal. So I think encouraging testing both of dams and sires is important. And then finally, you know, we kind of have to think about the carrot and the stick approach, kind of going through and scorched earth style, tossing everyone who carries the genetic disease won't benefit anybody. We're going to lose valuable genetic diversity. We're going to lose valuable genomes from horses who are excellent performers. And not to mention, we're going to lose a lot of members who are going to be burned in the process. So thinking about creative ways to incentivize making good long-term choices, like you know, cash incentives, year-end awards, or potentially some limits on the number of progeny from carrier stallions might potentially help things. And finally, if fragile foal does positively impact performance, and I have heard that there is some exciting work coming out of Europe that may show this, and actually we have a project on it as well that I'll talk a little bit about later. So if those research studies come out and document a performance advantage due to fragile foal, then we have a very, very careful situation to think about, in that we want to not only control that allele so that we can benefit from it as we can, but we need to think about how we select our animals positively. So can we be more specific than things like linear scoring, which I know starts like we're really only just getting comfortable with linear scoring, but linear scoring is still not very specific. And as we've seen in the livestock industry, having a consistent selection approach is far more effective. So we need to find a way to find excellent performers who do not have the alleles that are difficult to manage. Okay, I'm going to stop there just for a moment and address some questions. Do we have anyone who wants to speak up before I take questions from the chat? Nope. Great. I haven't lost anybody. You're all still there, right? Good. I do see some comments about impressive. He was a halter horse and he had HYPP. Is that why a lot of halter horses have it nowadays? You know, probably. Now, Impressive actually was a stallion who influenced other lines of quarter horses, but he's best known as a halter horse. And certainly if this allele impacts musculature, that particular body shape is most valued among the halter horses. There's probably two impacts there. And someone just said that they in fact have EDS and never realized how similar it was. Well, there you go. You know, so there's a perfect example of how you know, we need to think about how to live with the allele, not just how to necessarily eradicate it. And you probably have a fantastic extended trot. You just have never had an opportunity to show it off. So I'll look forward to that. That elasticity, I think, has a benefit. Wonderful. Okay, so let's think now about some complex traits. So one of the other topics that was high on the list was OCDs. And OCD is a story that is not simple, not simple at all. I'm going to come back to that next question in the chat too. So don't worry, Katie, I'm coming back to you, okay? All right. So let's go on this slight detour to talk about OCD. To get there, I'm going to take you to another disease, ironically enough. This is something called laryngeal neuropathy. And this is how I actually started to get down this particular path. Laryngeal neuropathy, or roaring, if you work with a lot of off-the-track thoroughbreds, you're no doubt familiar. Certainly a lot of our large horse breeds, they have that condition from time to time. And it's not unheard of at all among our warm bloods, although we do try to select for good airways. In this condition, it impacts one of the longest nerves in the body, the recurrent laryngeal nerve. For some sort of an unusual evolutionary throwback reason, that nerve takes a circuitous path down near your heart before it comes back up to your larynx, which is your voice box. Because it is particularly long, likely, it is prone to some neurodegenerative conditions, and in the horse, we see this as this recurrent laryngeal neuropathy. When that nerve begins to degenerate, the larynx itself loses its muscle control. If you're human, this means you start to lose your ability to speak or to sing. In horses, they lose the ability to open their airway in response to exercise and potentially close it tight when they swallow so that they don't inhale feed. It's this floppy larynx that causes that roaring sound that you hear in a horse who is working maximally and has the condition. The challenge here is that, like a lot of neurological conditions, we don't have a good treatment for it. Once that nerve function is lost, it's really tough to restore it. And we've tried some different things, a tie back to hold that open, but then they can potentially inhale feed, some electrical stimulation, all sorts of things, but it's really quite difficult. The interesting thing about RLN is that it has very different frequencies depending on what breed you're working with. In this case, the small breeds, ponies, are almost unknown to have the condition, whereas heavy horses, it's often estimated about 50%. Now, among our sport horses, I'll get to that, sorry, I'm getting ahead of myself, but the question I had here with the big horses and the small horses is that they have very different environments. They do very different things when they're in work. So it's rare to see Shetland ponies working at maximal effort. And our big horses tend to work fairly hard but slowly. So it's hard to say that because it's in different breeds, we will find a gene that's at different frequencies. In our sport horse, our light horse breeds, that frequency varies from between 11% almost as high as 42% depending on which population. So this is an aggregate of numbers coming from a collection of studies examining not just racing thoroughbreds, but also our sport horse warm bloods. So this is a condition that we have, and it can be a challenge. Less so for our dressage horses, except for those working at the very uppermost levels, certainly limiting for our event horses. Okay, so being geneticists, we brought out our great big hammer and we thought we would take a look. And this study was a result of a big collaboration we had while I was still up at Cornell. When we mapped the genes for recurrent laryngeal neuropathy, we discovered that, at least in the thoroughbred, it mapped very discreetly to chromosome 3. So you see my green box here? That's a mapping signal. Here we have the genome spread from the first chromosome on the left all the way to chromosome 32. And where you see the points go high on the graph, that's where we have strong signal for association. What was odd here is they actually called me into the collaboration after they had started doing the mapping along with Adam Boyko, who's a dog geneticist. And when they showed me this plot, I said, "You've mapped the wrong trait. We've already mapped this." Because we had just a year earlier published a study on height in the horse where we mapped it to the very same marker. Turns out it was no coincidence, because when we examined height in the same set of horses, we could again see a very strong signal. Now we have an interesting situation. So the same allele adds about 3 inches in height once you have between 0 copies, 1 copy, and 2 copy. And by the time you get to 2 copies, you have a 12-fold higher risk of developing recurrent laryngeal neuropathy. That is not a small risk, 12-fold. So if I said, you have a 12-fold higher risk of developing a heart condition if you do this, most of us would be willing to take health interventions to avoid that. So I think for horses, this is a place where we have some interventions. Right. So we mapped this for height in horses, not just in thoroughbreds, but across diverse breeds. And so this allele for height and for RLN can be found in many populations. And we very discreetly showed that it impacted specifically height and not necessarily other things like the thickness of their bone or specifically the length of the neck, although I want to go back and look at that with a more fine-tooth comb. What's interesting about height? Well, at least for the racing thoroughbred, where this allele is not infrequent, it is correlated to total earnings. Specifically, especially that's height in the front limbs. So horses who had longer limbs earned more money on the track. That means that there is positive selection for this allele in the thoroughbred, no doubt. Now, the other interesting thing is a few years after we started working on this region, we saw a nice paper from our collaborators there, actually in Germany, and Ottmar is still there working at the vet school, that showed mapping of OCD, osteochondrosis dissecans lesions, and again found the very same region of the genome. In this case, it was conferring 1.5 to 2 times the odds of having an OCD lesion in any of the 3 joints that they studied. This was the tarsus, the hock, and the fetlock. So the same gene is impacting 3 different traits that are all of economic importance to sport horses: height, a healthy airway, and healthy joints. It probably is positively selected for the impact on height, and then has mild negative selection in the occurrence of impatent airways and joint lesions that lead to arthritis. But I think that we have the opportunity here, now that we know what might be going on, to start to intervene. How might we intervene? Well, here's where I say we give this example of, we've looked at PSSM, we've looked at HYPP. The one common thing here might be growth. And so I'm hoping we can do some additional studies that would maybe you test your foals, and if they have this particular allele for height, you can begin to use some nutritional interventions to monitor their growth, to limit their growth, which we can see there are some fantastic studies done, particularly in the Selle Française, coming out of our colleagues in France, that show that if you feed your young horses differently, you get fewer OCD lesions. This isn't always practical. We need them to grow, and they need those calories so that they can start training. But for some individuals where the risk is higher, maybe you rebalance your cost to benefit ratio and change how you feed them. And myself and my nutritionist collaborators are literally chomping at the bit to do this study to see if we can identify the growth differences between those who have this allele and those who don't. All right, I can see the chat is starting to light up. So any live questions before I revisit some of our questions in the chat? No, that's okay. I like the chat. The chat's a great place for those of us who are shy to be able to speak up. So there's a question about, it's hard to eliminate PSSM because the allele frequency is very high. In the heavy horses, that is absolutely true. So how do you prevent further breeding of horses that have a chance for PSSM? Well, this is where you have to play the long-term ball game. And you find ways to strategically, selectively breed animals that have the traits that you particularly value, but don't carry this specific allele. In the Percheron horse, that's probably tough, although Percheron horses do fairly well, and most of them don't have a problem with tying up. So, some genetic background benefit can be found there, but, it's not going to happen overnight. You can't make big changes that are going to change a large allele frequency like that quickly, for sure. I won't have time to discuss vertebral fusion, but there are a couple spinal conditions that we are interested in. So if you send me an email later with some questions about vertebral fusion, I'll try to get to those. With PSSM2, we only have anecdotal stories suggesting it's not responding to the prescribed diet. And from those cases, I do not know what because once they discovered that I couldn't give them the answer with the genetic test, they often moved on to a new veterinarian or a nutritionist. So I think that's a key area. If we want to discover how to help these horses, we need more scientific research. Here's a question about, can you do surgery on a horse who is diagnosed with roaring? Well, I'm not a surgeon, but I did collaborate with surgeons up at Cornell. And the answer is yes, but that surgery has some drawbacks. Once that larynx is held open, they're at increased risk for inhaling food particles and developing things like pneumonia. And sometimes the surgery doesn't work. And sometimes the surgery can even make the condition worse. So that's something to very carefully discuss with your veterinary surgeon to decide if that's going to be the right choice for your horse. You probably should not do that before you have degeneration. Honestly, most horses, you don't even know they have the condition until you start to hear the noise, and that's where you already have some challenges with having being able to open their airway for exercise. Okay.
Can I ask something about
Sure.
your question? Because in the Netherlands, when young stallions are selected for the performance test or to go to the second round at the KWPN, then they are tested for that gene, for the roaring. And I know one young stallion that had it, and then they had surgery on him. But the question I have as well is then, if that stallion was diagnosed with it, does that mean that one of his parents is passing that gene on, or can it just all of a sudden appear in a certain cross, or is it something that's inheritable? Because why would they test it for the KWPN stallions if it's not inheritable?
Recurrent laryngeal neuropathy can result from accident. In this case, we studied the heritable form. Okay, so the nerve can be damaged by other things. One of the things my clinicians used to tell me is that they didn't recommend giving injections on the right-hand side of the neck because it is possible to accidentally strike that nerve, causing damage resulting in laryngeal neuropathy or paralysis. Okay, so there are the rare environmental thing that could happen, an accident that would not be heritable. However, that is extremely rare. It is very, very rare. Most horses who have recurrent laryngeal neuropathy, the vast majority of them in our study of thoroughbreds, carried this particular genetic defect or allele. Plus, if you want a tall horse, right? So the idea of giving him surgery when he was young and still standing him as a stallion, we have to think carefully about that, because he definitely, if he tested positive for this allele, it was the heritable form. If they used the laryngoscope or the fiber optic camera to look at his larynx and diagnosed in that way, it might have been the genetic form, or there's a small possibility it was an accident due to a bad injection. Very small possibility, right? So he almost certainly inherited that from his parents and certainly can give that to his offspring. It's an interesting case. Thank you for asking that question. Okay, let's move on. It's going to be a long night, so I want to get on to some more optimistic things, but fantastic discussions. I love to hear these types of unusual situations. All right, so now that we've kind of had the slightly depressing and definitely confusing section of the talk, I'm going to move on a little bit to something a little bit more optimistic. And as I have been in this career for a little while, I am starting actually to focus away from genetic diseases a bit and starting to look more at performance, and in particular, being a horse person. And being a horse person who, like all horse people, occasionally falls victim to the somewhat combustible combination of an intractable horse and the theory of gravity. I have that same appreciation for the value of a good temperament in a horse. So I have started to think more about how genetics may play a role in temperament. All right. So here is a case study provided to me by our friends at Edelon Genetics. You know, since they've started to do a whole lot of testing, they're definitely coming up on some more of these interesting situations. This was a jumper. I'm actually not going to show a photograph of him because while his owner was happy to have us use him as an example, he is such a visually recognizable horse. She didn't necessarily want to become instantly famous, at least among our genetics talk participants. But here's a horse, and if we look at his ancestry, I will definitely tell you he is a warm blood sport horse. And we can see that by looking at his genetic ancestry. This did not come from pedigrees. This came only from a hair sample. He carried about 42% thoroughbred, a little bit of what we call the carriage horse line, which I think gives you a well-set neck and a very expressive shoulder. He's got a little bit of heavy horse for durability, a little bit of Iberian and Arabian for some spice, and a touch of North Sea for, well, something we'll call tenacity. I don't know. This is very, very typical ancestry profile for a lot of our warm blood sport horse breeds. What was interesting about this horse is that he was submitted for comprehensive genetic testing with a history, and that he is a youth jumper. And by youth jumper, I mean a pretty serious jumper. He was in the 1.4 meter classes. So this horse can move, okay? But however, the owner was a little bit frustrated in that she had a hard time getting him to perform a good flying change. And he was sometimes very happy to trot in between the fences, which is impressive considering he was clearing 1.4 meters, but can be a little bit challenging for his rider, who would much rather have him canter smoothly between fences. What comprehensive genetic testing revealed is that he carried one copy of something called DMRT3, which is rather surprising to find in a warm blood sport horse. Not unheard of, but was surprising. And in this case, I think has something to do with his performance challenges. So let me explain why. DMRT3 is a little bit famous in our horse genetics world because it gained the cover of this journal called Nature. This is an Icelandic horse. He was used in the study where the allele was first observed. This is it's a bit like being on the cover of Rolling Stone if you were a horse geneticist. It was a big win for us. This particular gene is important for coordinating the firing of neurons involved in spinal circuits. And in this study, they found it very frequently among gaited horses. So those that do in the Icelandic horse, things like colt, they also found it in Tennessee walking horses and Missouri foxtrotters, and some of our passos. So at the time, they concluded that this had something to do with gaitedness, the ability to do intermediate gaits other than trot. So why is it in a sport horse who can clear 1.4 meters and clearly canters when he wants to? Let me give you a couple examples here of what it looks like. Edelon sent us some videos. This is a horse who does carry DMRT3, and as you can tell, he moves a little differently than our sport horse breeds. Here's another one. He does not carry DMRT3, and I think you can recognize there are some qualities to this trot that we definitely value in our sport horses. But both horses trot in some way. So clearly, DMRT3 is not as simple as they saw it in the original paper. Since then, we have also, through comprehensive genetic testing and assumption-free testing, we have observed it in all sorts of unusual places. For example, among Western pleasure horses. And in the pleasure horse world, there is no small controversy surrounding what they call a 4-beat lope. And they're trying to work on their judging standards to distinguish between a 4-beat lope, which is not considered desirable, and the proper 3-beat lope that they do necessarily want. But this is pretty hard to see. I know I have a hard time squinting at that picture. However, among those pleasure horses, we do see a significantly different frequency of DMRT3 compared to the background of general stock horses, suggesting that there is something valuable there. And probably some lines have used this DMRT3 to change the way their horse goes and are positively selecting for it positively. So in the case of our jumper horse, probably what it has changed is his ability to change into that canter is different than the other sport horses in the ring. And in his case, we might, rather than sending him for a neurological exam or putting him with a new trainer or trying a new type of martingale or bit or saddle or anything like that, I think we'll have to just understand that his particular behavioral idiosyncrasy is likely attributable to his genetics and is something that we'll have to work with and not work against. Okay, so that was a fun anecdote. This is one of my favorite quotes about the importance of the horse to human civilizations. We have worked with the horse in ways that is very different from all our other domesticated species. which has undoubtedly changed the way the horse thinks and behaves, maybe almost as much as it's changed the way that we might think and behave. So if there is any economic important trait in the horse, I would argue that behavior is definitely one of them. And in one way or another, we're all selecting for behavior when we breed our animals. So as a geneticist, what are our likely targets for things that impact behavior? In particular, you might be aware of the effect of a brain chemical called dopamine. So dopamine, it is a neurotransmitter. It's important for signaling between neurons, and it's released by the one end of a neuron and received by the other. And it's the target of many, many therapeutic drugs for humans with challenges with their psychological conditions. In dogs, it has particularly been variations in these genes have been associated with traits like aggression and anxiety. And in humans, there are polymorphisms in this particular set of genes that are associated with thrill-seeking behavior. I've always wanted to do a test looking at dopamine receptor genes in horses and their humans, because I would be curious to know if in our high-speed eventer types, if we see more thrill-seeking alleles in both the riders and the horses. But someday, if wishes were research grants, I'll get it done. In fact, based on this previous research, some colleagues in Japan launched a study to examine specifically one of these receptor genes called DRD4. This actually is not a recent study. They did this, a decade and a half ago, thinking very logically about where they could find impactful alleles. In their study, they found one specific change that had two forms, a G version and an A version. And of course, these horses, just like humans, have one copy of each. So horses that had two copies of the G were on average exhibiting behavioral tendencies that fit with the DRD4, what they called a trend in curiosity. And horses that had one or two copies of the A version had a set of behaviors that they categorized as being more vigilant and more cautious. Now, I think it's interesting to note that there was still quite a bit of polymorphism for these two types of temperaments, because I think depending on the use of the horse, we value them for very different things. If I was out looking to purchase a new 4-H horse for my daughter, I would definitely consider looking for horses that had the GG form. A little more curious, a little more bold, maybe less likely to exhibit some ill-timed fear behaviors. But if you're looking for a trail horse or a race horse, you might prefer the AG or AA genotype, which might render this horse to be a little bit more quick to be on the lookout and maybe faster on their reflexes. Further studies will tell us for sure, but I do think this is something that could be very interesting to apply broader in our breeding program. Now, one more recent study in 2013 did illustrate a potential health challenge here, in that horses who had at least one A copy, so that's what this notation means, had one A and then the other could be either A or G, that they did exhibit more frustration when stabled. And frustration type behaviors might then lead to things like stall walking and weaving and cribbing if they contribute to the anxiety and stress that is a part of the environmental component to those conditions. So just sort of emphasizes the general tendencies that these horses have, and maybe you all know that kind of type A horse who's always a little bit worried to be in the stall, but darn quick on the stadium jumping course. So here we might be able to inform our management with this allele as well. So given the importance that I attribute to this particular allele, I want to continue the work by looking at some of our ongoing studies. And this really raised a pretty big question for us, that aside from the obvious combination here of gravity and some interesting life choices, how do we define these qualities, and then how do we measure them scientifically? And these are some very, very difficult questions. We have different types of riders, we have different riding disciplines. They value different things. And then how do I measure them consistently and scientifically so I can have some successful studies? So this is the experimental setup that we have started with, and we are starting very, very simple. You know, there's always a combination of genetics and environment, and so we tried to target something where we could control the environment by using our UF horses on our UF farm, and by breaking some of these behaviors down into their most basic components. So in this experiment, we have some key pieces of equipment. The umbrella here that you see on the left is a standard automatic umbrella that opens with the push of a button. We always use the same color, and they all get a lot of WD-40, so they all open quickly. We train the horse to enter the standard round pen on its own and to proceed from a start box to a feed pan where they get a grain treat. And these horses, they become professionals at this activity. Most of them can walk themselves from that box directly to the pan, no problem at all. So we've tried to subtract the human as much as possible out of this particular test. We measure their reaction to that umbrella in two key ways. So we are always expanding our scientific equipment here. First, their gross behaviors based on video analysis that's categorized very carefully in something called an ethogram. And then specifically, we're looking at the cardiac startle response. So this is the subconscious part of that fear or that startle, right? So their heart will register and prepare for the need for fight or flight before your brain has a chance to consciously make a decision. So we're looking at it two different levels here. So this video is going to show you in a more high resolution version how this proceeds, and it's pretty predictable. I feel like I've had this happen at the side of my dressage ring in the middle of my test once or twice when it starts raining and some ill-meaning parent or oblivious parent just opens their umbrella without meaning to totally derail my day. But as you can see, these horses react fairly predictably. What's interesting is that after doing this experiment for 6 years and well over 100 UF stock horses, we can start to see an interesting breadth of response. So come on, little horse, let's go. Here we go. So here's some actual experimental footage of younger horses. This is a weanling. We do it at weaning age, and then we do it at 2 years of age once they're under saddle and in training. This little feller is famous. He's one of our lowest scores on our startle scale. He behaved very similarly as a 2-year-old, and as a 3-year-old took his young owner to the state 4-H finals here in Florida and did things like trail classes as a 3-year-old. No problem at all. Now, some of his cohort behave very differently. Here's another example. This little horse wanted nothing to do with that pan, and some of them want nothing to do with umbrellas. We actually do some desensitization at the end of this experiment because we don't want to traumatize some horses to umbrellas, right? So by working at this from a scientific perspective, we're starting to be able to assemble something that we can look at the genetics for. All right, one more quiz. As horse people, as horse breeders, how much of that spook do you think is genetic? You think about 0% of that response is genetic. It's all environment. It's all training. We can fix everything. I should just give up. Sometimes I feel that way anyways. It's 20% genetic, 80% environmental. It's 60% genetic, 40% environment. It was a windy day. It was cold. Cows were mooing next door, and that happens. Or it's 100% genetic. They're bred this way. They hit the ground. They're spooky from day one. Install the seatbelt on the saddle and hope for the best. All right. I see Katie says 20%. I saw some heads nodding at 20%. Yeah. Okay. Well, our preliminary data shows, ding, 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 for the most basic component, that very first response is about, right now, our estimates are 60% genetic. So what they do in the first 5 to 10 seconds, very genetic. I will say on the flip side that the other components, the more gross motor components, what happens from 10 seconds on, when we measure the genetics, that's nothing, virtually nothing. So our hypothesis is at this point is that the predisposition to the startle reflex is stronger in some horses than others. other horses. And then the environment and our training, what they have learned, alters how they handle that initial startle reflex, explaining why we don't see genetic influence for 10 seconds and beyond, which I think is pretty cool, because that means two things. One, we can definitely, I think, develop tools to better understand the biology of fear and startle and how it might impact management. Are these horses going to be more likely to crib? Are they going to be more likely to get ulcers? Are they going to be better predisposed to being racehorses and jumpers? And can we identify horses that have this predisposition, change their management, change their training, and be able to get them to the disciplines where they are best suited and can excel by playing on their strengths? This is my hope. Anyhow, I have a PhD student currently on the project, and we are hoping to get some funding from groups like NSF, National Science Foundation. This raises an additional question, though, like how else can we manage our potential? And so here I'm going to circle back just a little bit to some of our fragile foal challenges. Okay, and this is part of the study is done primarily by my other student, a master's student. We have in the sport horse world this phenomena of the veterinary inspection. So at FEI levels, we undergo a veterinary inspection at many of our different competitions. And to those of us who are scientists, having gone to these for many years for the enjoyment of seeing all the crazy outfits, it kind of dawned on me that this is a fantastic model that we aren't necessarily taking advantage of. So we're working now to develop artificial intelligence tools to label skeletal landmarks on these horses and extract variables relevant to performance. In particular, we're interested in joint flexion so that we can examine correlation to alleles like fragile foal. That's one of our early goals right now. It's important to note that these labels are entirely applied by the computer. We have right now in our library probably, I forget, 1,200, 1,400 videos from FEI veterinary inspections. And we're going to start to build a very sizable database cataloging the movement of horses at the upper international levels of the sport horse world. Mostly eventers so far because they give us 2 jogs, 2 opportunities, but also dressage, show jumpers, and other FEI disciplines. Here's another example. This is a horse. This is actually a Hanoverian mare who was competing at the 4-star level here in Florida. The video is a bit broader. You can see an odd sort of shape in the back. That is where we're trying to track the rider or the handler's legs so we can subtract them from the equation. What's nice about this particular tool is that while it's useful to me as a geneticist to look at things like gait quality, elasticity, fatigue. Right now we're looking at fatigue in horses prior to and after cross country to look at differences in movement specific to dressage trained horses versus show jumpers. Is I hope the tool will have long-term applications in things like selection and identification of lameness. But it's still very much in our preliminary phases. But for right now, we're starting off. We will be examining fragile foal in horses where we can measure joint flexibility in their FEI jogs. It's not a simple process, and so I'll fly through this. We're using some AI-based software that was developed for other animals. And the FEI model is fantastic because we're able to get a very large sample size, which is unusual for horse studies because we're generally limited in resources and the availability of these large animals. We're going to develop some complex models mathematically. We're working on that part now, particularly in collaboration with our colleagues at Auburn. And eventually, I want to be able to compare these data to these horses' genomes to identify which regions are particularly impactful in athletic performance, more on the musculoskeletal side, whereas my earlier research is more focused on maybe the mental side, which is no small component when it comes to our sport horses. As all of you know, if they don't want to do it, they don't do it. Not at all. All right. Those projects, they are underway, and we are challenged with being able to find funding for the type of work that is relevant for our horse industry. USDA is dubious that the horse is an agricultural activity. NIH isn't sure that they're a good biomedical model. and NSF is not convinced yet that we have the right model. So if you have your cell phone out, snap a quick pic and consider helping us to raise some money for those 2 studies. All right. So I am going to address a couple questions from the chat. One question is there a correlation between kissing spines and PSSM? That is a great question. I am a collaborator on another study specifically examining PSSM. And in preliminary data that we presented at the AAEP conference in December, we do not see an overlap in the genetic signal for kissing spine and that for PSSM. So that's an interesting hypothesis. I don't have any good evidence for it yet, but I'll definitely keep it in mind. So here we've got a question about how do we see our breeding decisions being made for 1 year from now, 5 years from now, and beyond. And actually, I want to talk a little bit more about some of our other topics before we come back to that. I think it's an extremely important question, and I hope that at the end of this talk, when you've seen some of the available genetic tools, you might have a slightly different answer than you had at the beginning of the talk. And definitely lots of surprise at that initial startle response. Yeah, it's really neat. I think that one reason we're starting to really pick it apart is because we've been very careful in how we are running our experiment. But certainly there have been lots of rumors. You know, they used to say that the storm cat lines and thoroughbreds were known for being very intractable. You know, we've kind of heard these things for years. So I think there's some horse person's wisdom there. Carolyn asked, is it possible that there's some impact because they're prey animals? And I would say absolutely. And it's one argument I'm making in our grants now is that the horse is one of the few prey animals that has this close interspecies relationship with humans. The next one that we might call a corollary to that would be dogs. We have a big vocabulary with our dogs. We can communicate with them very well. Think about a working sheepdog. With our horses, we have a very big vocabulary, but we probably didn't eat dogs as often as we ate humans prehistorically, based on the skeletons we find in middens and things like that. So the relationship between the horses and humans has probably changed more drastically. And their instincts as far as avoiding predation is probably very different than those of the dog. So my hypothesis here is that their domestication history is quite unique and makes them very interesting to study. And one more note from Laura St. Clair. She wanted us to mention that Edelon has offered discount codes on some packages. So we can follow up with that at the end too. And I will mention some other collaborations with Edelon and other testing companies as I can. In particular, the kissing spine study I just mentioned was presented at AAEP. That was a collaboration with Edelon and a number of their veterinary advisors. So it's where we're starting to try to take advantage of some citizen science approaches to gather large collections of data that are needed to do the work. Okay. Any questions from the audience before I move on to our last topic? Try to get us out of here before it gets too late. Is that a frustrated question? No.
No, just on. Can you hear me?
I can hear you.
Okay. It'd be easier for me to ask this way. The relationship between the horse and the human as compared to the dog and the human. The horse and the human is so different because it's prey predator. And that was my thing about, like, because of the genetics, does say like a rabbit have the same like that genetic code because they're prey animals also? I mean, that was just my thought.
Right.
And because like I said, the relationship is prey predator as compared to human dog, which is predator predator.
Right.
Or even like my horses have relationships with my neighbor's goats, you know, and they're prey. So I was just wondering, because it is such an odd relationship between horse and human.
Well,
that was kind of my thought, and I couldn't type it fast enough.
Sure. I agree with you. And that's one of my arguments for why it's important to study this particular relationship, because it is unique in domestication history. So our cattle, our goats, our sheep, our rabbits, those were predators and their prey, right? But with those other livestock animals, we do not have this complex network of interspecies communication that results in our ability to cooperate with them to be able to go and jump, 6-foot puissance fences or maintain the movements of Grand Prix dressage. That communication part, I would argue, we don't have with dairy cattle or with our pet rabbits.
True.
So I think I would expect we'll find some different changes in the horse that we won't see in any other animal, and that those changes will highlight some pathways in the brain that will be important for better understanding brain development and potentially brain pathologies even in other species. So at least that's what I keep telling the grant reviewers. I'll keep trying.
Thank you.
So, all right. So let's go one more round around to the other thing that happens commonly in domestication: changes in coat color. So the thing I'm going to highlight here is, color is something, in the sport horse world, we kind of think of it superficially, like it's a nice thing. We have our favorite colors, but at the end of the day, we want to be sure they perform well, right? But sometimes these colors have important implications beyond just what's on the surface. So color is something that no doubt captured our fascination early on in our relationship with horses. And even some of these 20,000-year-old cave paintings document some interesting variations in color here. Recently, we've used DNA to actually document that the genes that create this, what we call the leopard complex, but what here in the States is known often as Appaloosa coat color, actually those alleles date back 12,000 years. So it's not impossible that the spots that we see on those cave painting horses were actually what that observer saw in real life. This is particularly interesting. I don't go into the genes that create the Appaloosa or LP leopard complex pattern. It's also, of course, in our Knabstruper horses. But one of those genes, TRPM1, is associated with a condition called congenital stationary night blindness. So horses who have 2 copies of that gene are blind in low light levels, which would strike me as being a disadvantage if you're likely to be hunted by things like saber-toothed cats. So some of these alleles are quite old, and we have to wonder what was sustaining them at that time. In the case of Appaloosa, we kind of joke that it might have been camouflage, that they might have been easily hidden in the Ice Age in the snow piles. But we can see a sudden emergence of many more coat color alleles in these prehistoric samples. In particular, we document that this bright pattern called Sabino 1, which is very most frequently found here in the States in the Tennessee walking horse, but is documented in a number of breeds. That's the oldest white spotting pattern. It was first observed, to our knowledge, in a single horse that died on the Mongolian steppe 4,000 years ago. And one of our more basic alleles, the difference between black and bay, an ASIP allele, that was seen almost 6,000 years ago. So there are some environmental rationales for why these colors might be different. So here again, we have another cave artist who might be showing us the real, they're a more realistic picture than we thought they did. So darker wolves are hypothesized to be better suited to hunting in forests, and we might suggest that some of these colors are due to different ecosystems in which these horses might have lived. But this rapid expansion correlating with this time period also matches well with when we think we started to have a horse-human relationship, and our horses might have been positively selected for color strictly because we valued it and not because it created some difference in their fitness. Okay, so since we've started selecting for color, we have a number of alleles, and this is a very, very short list. I started with some of the oldest ones that we've observed. But the difficulty is not that some of them do have important health impacts, and these bear keeping in mind when we are working with them as breeders. So I'm going to cover a couple of them here as we move on through the slides. Here's an example. We're in frame overo, of course, is a cornerstone pattern in breeds like the American paint horse, but it is found rarely in our warm blood sport horse breeds and even among thoroughbreds, very rarely. What's neat about frame overo is it is, as a spotting pattern, dominant, but it has a diverse degree of expression, so that some horses have very loud patterns like the horse on the left, and other horses have very mild or more subtle patterns like the horse on the right. That makes it a little bit tough to recognize by looking only at the skin. The downside, especially with horses who have the milder expression of the pattern, comes a little later on. Just to review, here's our dominant illustration for that single allele of the overo pattern. That means one sire or parent has one allele. If the other parent has no alleles, we'll see that allele passed on to the offspring at a rate of about 50% or half the time. I see lots of people who have experience with overo. So as those folks know, the downside to frame overo is that this gene has more than one job. In the skin, it's responsible for directing the migration of pigment cells across the embryo. But in the gut, it's responsible for helping to populate the gut with some important nerves that direct gut motility. So horses who are missing two copies of this functional gene, so the overo change renders it dysfunctional. So if they have two copies of the overo gene, they do not have normal gut motility and will die shortly after birth from a fairly catastrophic colic. As you can see from the foal in this picture, they also tend to be entirely white, whereas one copy only partially impairs pigment cell migration. Two copies very severely impairs pigment cell migration. So we say that the disease, although it's the very same allele, we say that the disease itself is recessive because it takes two copies to observe it. With one copy, which we see in half of this hypothetical foal crop, you get the valuable spotting pattern, but with two copies, we get a very terrible and lethal condition. I call that recessive. This is just one example of how coat colors often have dual roles. Those alleles can be important in two ways. Here's another example, and my PowerPoint has gone into a bit of its own sort of lack of innovation here and has thrown a picture up where it doesn't belong. But what I'm showing you here is another recessive allele is chestnut. So that is very commonly found in our sport horses. And I'm getting to our comment about a farrier here who had some wisdom on the subject. So chestnut, we say it's recessive because what you can't see, there's two black horses here, and if we called them two parents, they could potentially have a chestnut foal. The recessive allele itself is due to a change in the melanocortin-1 receptor. Its symbol is MC1R, and that gene functions like a light switch. So the graphic here in the center is the illustration of a melanocyte, and the melanocytes can make two types of pigment in people and in horses, a reddish type of color and a black pigment. And MC1R is the light switch that determines which one the cell makes, reddish color or black color. When the light switch is broken, like this E allele change creates, the horse can only make the red color. Black horses are actually both black and red. It's just the black pigment is the stronger color that we see on the surface. As we might be aware, there is some conventional sort of colloquial horseman's wisdom with regards to coat color and behavior. This is a little poem. I never have been able to figure out what the attribution was, but it says here in the middle, a bay is hardy, but a chestnut is fast, and a gray is gentle, and a sorrel is hot. And so this rumor predates the rapid wildfire sort of meme style internet spread of rumors that we see today, no doubt. And in fact, as the rider of an off-the-track thoroughbred mare, this is actually the back of my truck for a number of years until that truck finally had to go. And actually, I have another version of that bumper sticker right here in my office. I have sort of lived this effect. So why might that be? Well, as it turns out, we can learn a lot about the function of this receptor based on work done in humans. So MC1R in the skin functions as that light switch, but in some of the neurons important for pain perception, it has a very different function. So when that light switch is broken, you get only red production and you get altered function in the pain perception pathways. So humans who have MC1R changes that are somewhat similar to the chestnut allele in horses specifically get less relief from analgesic drugs. They have increased sensitivity to thermal pain, and they have a need for additional anesthetics in order to undergo typical medical procedures. Which brings me to another study that someday when I win the lottery I will do, is I have a strong suspicion that our chestnut horses have a similar effect, that they may require additional analgesia when undergoing medical procedures, and they may be somewhat more sensitive when in training, because many of the things that a bay horse would not find uncomfortable, a redhead might, humans and horses. So keep that in mind. If you have a hot little chestnut filly at home, give her a little bit of slack, because it's possible the world is just that much more irritating to her, and give her some special treatment. Okay, now we have some scientific documentation to this, although I do think the case is still open. This study looked at over, gosh, they almost got to 500 in the total study, but they're looking at a diverse set of breeds, and they used photographs to classify the horses chestnut or non-chestnut. And they found differences in specific survey questions answered by the horse's owner. So interestingly, they found that chestnut horses were more likely to have a positive answer to the question, does the horse allow its feet to be picked up by a stranger? But the answers are more likely to be negative to survey questions like, how does the horse approach familiar stationary objects, and how does the horse approach unfamiliar animals? So these suggest a broader behavioral trend that may stem from differences in pain tolerance, but certainly impact behaviors that are very practical on the farm. It was a neat study, but I think that the photograph was potentially a limitation. And if you want to read it, there's the reference. You can certainly look it up. So we did a similar study with a much smaller sample set, looking only at Tennessee walking horses, because my PhD student at the time had a special interest in Tennessee walking horses. And one of the first things that we discovered is it was quite difficult to attribute color based on photographs. We actually were failing at it quite miserably. So we fell back on our genetics and we determined the alleles for each of the horses in our study. Now, as it turns out, chestnuts are pretty hard to find among Tennessee walking horses, but we did identify a number of black horses. And in comparing them to owner-reported temperament surveys, we discovered that our black horses had a significantly higher score for a temperament trait we called self-reliance. So these animals, particularly when they were mares, tended to be more willing to be on their own than horses of other colors. Oops, my annotations are popping on there for you. This brings us to a second gene in this system, ASIP. So ASIP creates the difference between bay and black. That was the allele I mentioned a while ago as one of the first novel coat colors we observed around the time of domestication about 6,000 years ago. ASIP also has a multi-function sort of toolkit to it here. It functions not only in pigmentation, as we can see in this array of multicolor mice, but also in lipid metabolism and regulating the metabolism of those fats in adipocytes, which are fat cells. In this example, we have an allele called agouti yellow, and the effect on the color and obesity due to lipid metabolism, you can kind of see in the spectrum. I like to call these little pumpkin mice. They'd be sort of the lighter color, and then the darker bay mice will be down here on the right. They have the same allele. The key difference here was in what their mother was fed during gestation. So these two mice here, just to show you the extremes, they have the same genotype, but this genotype confers a specific response to maternal diet. In this case, it's supplementation with methionine, which many of you are familiar with as a horse supplement as well. These mice, when they have the adult onset obesity phenotype resulting from the interaction between maternal diet and this allele, have many of the same health detriments that adult onset obesity has in humans and potentially has in horses. So to study this a little bit more closely, we launched a research project using recycled data from other studies where we examined the darkness of horses who carried one bay allele. So these are not black on the right-hand side, they are still bay. But we observed considerable variation, not only in our UF quarter horses, which you see arrayed across the bottom, but also we included Arabian and Persian horses because we had leftover data from some other studies there. The allele that we discovered changed their color from light with single copies here to a medium color to a dark color, but there was a lot of variation within genotypes. And I'm very curious to see if in the future we can create a research study that will examine maternal diet impacts on individual genotypes within this series, because the allele that we discovered was very significant, but not the whole story. It brings me back a little bit to OCD and those studies that were done in France looking at sport horses, because they noticed differences not just in the diet that was fed the young horses themselves, but also in the diets and the obesity rates among the mothers of those foals with obesity. So I think there's a lot here that our coat color molecular mechanisms have left to teach us about cell signaling and health and management of our young horses. Okay, one more quiz, and then I'm going to address the questions in the chat. Since we're talking about coat colors and health challenges, and we've already heard some farriers' wisdom, are white feet weaker than dark colored feet? We can shake our heads. No, we can shake our heads. Yes, what do we think? Yeah, so some of us have noticed no difference. The science tends to agree, and I've got a link here. I can share the study with you. When we look mechanically at the strength of the hoof horn itself, we see no difference. Here, the color change results only from the deposition of melanin. I will say that where this myth might have originated is that we do see white markings more frequently in light horse breeds than we see in heavy horses. And we do also see differences in hoof horn thickness between light horses and heavy horses. So we may have had a sort of a correlation that between frequency of white markings and the thickness of hoof horn that was due to general breed trends, but wasn't actually a causative relationship. Science likes to do that to us from time to time, convinces us there's correlation does not equal causation. So I'd like to, let's review what we've gone over, and then I'll get through some of these really fascinating questions in the chat. And I may have to turn my light on. As you can see, it's gotten dark here in Florida. Overall, what I want you to remember are 3 key points. First of all, we can and should test for many health and management relevant traits, and some of those are closely tied to performance. So we have the opportunity to benefit from both sides of the coin there. We can better manage our health conditions, but we also can improve the things that we find valuable in our animals. I want you to keep in mind that genes do influence things like behavior and performance, and in some cases, they can have a very large influence. Not all by any means, but for some specific horses, it's a very large component of their equation. Think about the horse with pain from PSSM that was causing him to buck, right? So if you have a behavioral issue or challenge with performance, don't forget to think a little bit about genetics. And remember that things as superficial as color can still be economically valuable, even in breeds that don't have color specifically in their breed standard. The colors are still part of biology, and biology will have something relevant to consider. All right, so let me address some of the questions here, and then I'll tell you a bit about our class. Yes, so some of our audience mentioned that things like domestication syndrome and the example of our domesticated foxes that developed changes in things like their fur color, floppy ears, and their overall demeanor as part of as the overall change in their phenotype as they went from a wild-derived population to a domesticated one. In some cases, these changes are concurrent because they might be impacting the same pathways in the nervous system. But in other situations, they probably were coincidental. And I think understanding those relationships is what makes studying our domesticated species such a fascinating model in developmental biology. We've got a couple folks out there who have Overos, who have the lethal white gene. You know, it's a fantastic color. My daughter's current 4-H horse has the lethal white Overo gene and no spotting pattern whatsoever to speak of. So we don't plan on breeding her, but had she been sold as a breeding horse, it could have been a potentially serious surprise had we not had her genetic tested. And someone here has noticed that our paint horses tend to color code themselves in the pasture, grouping themselves by similar colors. You know, that's interesting. Maybe they have similar temperaments if they're chestnut and non-chestnut. I don't know. Maybe they're related to one another. We certainly know that in the wild, horses do react differently in their social groups, depending on whether they're reacting with relatives or non-relatives. And relatives would be more likely to share color alleles. So that's kind of interesting. We'll have to look at that. Oh, and somebody says they're afraid to breed. Oh, don't be afraid. Don't be afraid. You know, these things, the thing I want to point out is that all of these effects have always been there, always been there. The difference is we were just ignorant, right? We just didn't know. So we can kind of take the ostrich route and stick our heads in the sand and kind of pretend we didn't hear about all this, or we just use this knowledge to give us some power, learn a little bit more. And I think as an industry, we really are going to have to start to breed smarter rather than harder, to be a little bit more strategic. Not too difficult, I promise. And we've got lots of folks out there to try to help you now. 10 years ago, you didn't have nearly the number of people who are educated in horse genetics to be able to help to guide you through the process. So reach out, get some help, and, you know, keep reading, because I think that on the positive side, you have some tools that will enable you to do things that you've always wished you could accomplish, but you weren't able to do so. Not all of them, but some of them. Okay. One way to address your fear is to learn more. And I do have an online horse genetics class that is aimed at the horse breeder. It does talk about a lot of biology, but I think knowing the biology helps you to keep in mind where you can best apply these genetic alleles in your own breeding program. We have switched it to rolling admission. That's great because you can join the class at any time. The downside is that sometimes we have a lot of students, sometimes we have few students. And so the interactive pieces are sometimes too quiet and sometimes a little awkward. So we're still kind of fiddling with our online model to see how we can get that to work. We have 2 courses in a series of 3, and the third, I promise I will finish soon. I've been working on it for, quite a while. COVID derailed that just a bit. Okay, with that, if we have any other remaining questions, I'll be happy to take those. And I'm going to go turn the light on so you can see me. Any more questions? Anything about hoof color or some of the biology involved?
And I want to mention quickly, I want to mention really quickly too, before people, before we get into questions, that we do have some discount codes that I can post. And Dr. Brooks, if you could mention a little bit about the work that you do with the different testing companies and kind of how you work with the different companies and so forth, that would be really helpful as well.
Well, I do collaborate with several different groups. Being that we're here in the state of Florida, I don't endorse any one testing company by any means. I think that you really got to look at the different offerings and see what provides you the best value and the best customer service and the most accurate information. In some situations, I've been involved in cases where we've had issues with some companies who were using tests that were not what was originally designed by the scientists to try to avoid patents and things, and that has created some court cases that were rather awkward. So make sure that your company is using the right tests, and I like to see them publishing scientifically and be members of the ISAG, International Society for Animal Genetics. But we're trying to branch out. As you probably have noticed, I've mentioned a couple of times that finding funding for horse research is very difficult. And that's not unique to genetics. That's true for all of our horse research disciplines in reproduction and nutrition. So one of the routes that we're starting to explore more are these citizen science and public-private partnership approaches to getting some of our research done. You know, a lot of my research has always focused on partnering with horse owners and getting them to send in samples and maybe photos for phenotypes or measurements or data from their veterinarian. But when we partner with a commercial company, it gives us the opportunity for them to be able to get information that's usable right now and potentially contribute information that can be usable in the future, especially when it's combined with information from many hundreds of horse owners. So I mentioned at some point some of the studies that were done. You know, Etalon is nice because they freely give me data to be able to use in my talks. Some of the other companies are a little bit more shy about that. But you know, we've collaborated with a number of other groups in the U.S. and in Europe on several different things. I think one of the most exciting ones right now is kissing spines because it is so common in our thoroughbred and our sport horse breeds, and it is particularly frustrating, and it has a behavioral component because often we don't notice this orthopedic condition because the horse is visibly lame. We usually notice it because they have a behavioral challenge that is resulting from the pain that they're experiencing. And maybe those chestnuts are experiencing more pain, right? That one is a classic citizen science type model where horse owners are sending in radiographs from their veterinarians. They're getting some genetic testing done as part of the partnership, and then their data is going together into one big study so that we can hope to understand some of the genetic component to kissing spines.
Right, can I ask a question? Thanks. Kevin Lewis from Benchmark Hanoverians in Southeast Queensland here. How are you going over there in USA?
We're doing great. So glad to hear you're joining us from the other side of the world.
Thank you. No worries. We're always interested in genetics. Just a quick question about we've got a 2-year-old filly who is by it's a Hanoverian from Europe, and we were concerned that maybe it had a problem with PSSM. We tried to find a genetics lab in Australia to test it. We couldn't find one. Germany wouldn't do it for us, so we sent it to a genetics lab in America, and they did it, and it came back positive. And it cost us $500, which I don't know whether you think that's a lot of money, but when you've got a herd of mares, it's fairly expensive. And it was interesting to know when you said there's no verifiable genetic test for PSSM2. What confuses me a little bit. It come back positive. Is there no correlation between what we see on the ground and what actually the genetic test shows?
So you tested for PSSM type 1?
Well, it came back with a number of there was a number of ones. 2 actually come back positive to 2. Yeah.
Okay, so the PSSM type 1 test is very, very well validated. That one, we know the mechanism that generates the disease fairly well. It's been studied for a number of years. PSSM type 1 is a very, very good genetic test. PSSM type 2, when it's studied in a population of animals, has no statistical correlation to the disease itself. That doesn't mean it isn't occasionally positive in animals that do have disease. It just means that, you know, if you look at a number of them, that relationship starts to fall apart. It's unfortunate that she spent a lot of time and effort and money getting the PSSM type 2 tests run. Because I don't think in this case it has actually given you much information as to how frequently that horse might pass on PSSM2 to the next generation, or how you might treat it. If the horse does have clinical PSSM based on the biopsy, muscle biopsy, that does still suggest that even if they're PSSM type 1 negative, that you should consult with your veterinarian and your nutritionist for sure. But in most cases, they'll say try the PSSM diet because it might work and it's easy to implement. So I'm very, very sorry that you spent a lot of money. And if you're interested, if you send me an email, I can send you the study on the PSSM type 2 test that was recently completed by some of our colleagues here in the States that show that it does not have a good statistical correlation. It does not predict the disease itself.
Okay, so it might show up sort of early, later in life, sort of thing with the mare.
So she's positive. So you got a positive result for PSSM type 2, but she doesn't have clinical signs.
She doesn't seem to have it at this stage. She's been broken in and worked lightly, but doesn't seem to have any problem.
Yeah, I think you could rest fairly easy then, because that PSSM type 2 genetic test doesn't actually correlate with disease. So hopefully that means that actually she'll have a happy, healthy, athletic life without any PSSM disease.
Okay. Oh, well,
Good
the
news,
jury's out with me, but I'll just see what happens. Yeah.
Okay. Well, definitely feel free to email me, and I'll be happy to send you some of the studies and things about the PSSM type 2. And if you want to if you would like me to give you a more personalized opinion, I'm happy to look at genotypes as well. Let's look at some of the other questions here. That's definitely a very interesting one. One is on the genetics of belly spots and warm bloods. I actually had a slide on that, and I was worried we were going to go over time, so I took it out. But there are a number of alleles that contribute to things like belly spots, including one called W20, which is present in warm bloods. It tends to be due to multiple alleles and not just single alleles most of the time, but not always. So in some cases you can test for those, and in other cases it's going to be 5 or 6 alleles, that some of which are testable and some of them are not, and the additive model will be pretty complex. So belly spots and warm bloods is fun. The other thing to remember is that the migration of those melanocytes across the surface of the skin is partially due to random chance. So just like our frame ovaros, having one copy of that overo allele doesn't guarantee you get a bright pattern. Sometimes you get very little patterning. And just like when you throw paint against a wall, there's some entropy to where it lands, some chaos to it, right? So in some warm bloods with belly spots, they have the typical Sox and Blaze type genes, but they got lucky and the melanocytes landed in a different place and you got a little extra color. We have another question here about Rabicano. We have a study on Rabicano, and I see that my former student, Laura Patterson-Rosa, is in the audience. We didn't have enough funding to continue the Rabicano project, so Laura is going to take the Rabicano project with her and hopefully get to complete that as part of her ongoing adventures. So if you have a Rabicano horse and are interested, contact us and we can add you to that study. My own suspicion is that what we call Rabicano is actually due to some different alleles. I think just like for the dominant white type patterns, we're going to find different alleles in different breeds of horses creating similar phenotypes. So here's another interesting question. What are the genetics of a liver chestnut versus a regular chestnut? Well, We actually just this summer, I have the proofs in my inbox. I need to finish that, but we're in the process of publishing a paper on a genetic locus that contributes to the differences between liver chestnut and regular chestnut. So that's something we'll be able to add, another genetic allele that you can have tested and can predict that. This gene is kind of interesting because we have no idea what it does in any species, but we think it binds with another protein that creates a type of albinism in people. So it's going to be a new addition to the collection of genes that are in the pathway for pigmentation. So we'll get that paper out hopefully soon if I get those proofs turned in. And if you're interested in that, we'll be able to talk about that a little bit more next time. Okay, and then Dr. Rosa wanted me to also mention one of our other recent publications that was on anhydrosis. So this is dry coat or non-sweaters, another fascinating topic. I strongly encourage you to invite Dr. Rosa to give a talk one night. Her part of her dissertation was on mapping locus for anhydrosis that's important for regulating ions. So this particular allele confers risk for developing what we call congenital idiopathic anhydrosis. So horses that will develop they lose the ability to sweat over time for reasons unrelated to things like electrolyte imbalance and diet. So that does impact a lot of our sport horses, especially in hot climates like we have here in Florida. So I'm sure she'll be excited to talk about that very recent discovery very soon. Right, so is there any questions I have missed?
Everybody good? This has been excellent. I thank you so much. It's really it's just an incredible area, and you've opened our eyes to so many new applications of it, and you've also reassured us in so many ways. I think a lot of us will be more comfortable moving forward and really investigating our horses at a whole new level. It's a very exciting time. Thank you very much.
Great. So glad to for the invitation, and I love getting to talk to everyone.